welcome to our sixth podcast in the information domain. In this one, we are going to focus on how the cell cycle is controlled. So how do we control cell division? Because in the last one we mentioned that that is, something I wanted you guys to think about as to why is it so vital that the ability to create more cells be so regulated. And we're going to bring into this discussion the impact of what cancer is and why it happens. Okay, so why? Why should cells only divide when they are in essence told to or when they're needed to? Okay, that's kind of an important thing to think about. Okay, because why should they? And then they should, because if things get out of control, you're going to wind up with cells not working quite the way that they're supposed to. They might lose their ability to function in the way that they're supposed to. The idea that most of the cell's life should be spent in interphase, doing the job that it needs to, and there are various checkpoints as it moves through the cell cycle that help it work its way to know when should it move into, let's say from G1 to S, when should it move into mitosis. So these checkpoints are vital for the continuation in that control that the cell is going to need to do. So there's a point in G1, there's a point in G2, and there's a point in mitosis when it actually happens. So it's good to think of it with these checkpoints that the cell has to be able to, can it pass? In order to pass through to the next level, there are certain things that need to be happening. All right, so in essence, when you have something like a breast cancer cell, it's becoming a selfish cell. It doesn't do the job it's supposed to anymore. It is focused primarily on only dividing, only being sort of sucked into that state of going around, and it loses its ability to do the original job that it had. So at the G1 checkpoint, this determines whether or not we should move into S. Okay, so it either does or it doesn't. If it doesn't pass the checkpoint, it's going to move into G0, in which case it's going to just live out its life and go in with what we've called senescent cells and never divide again. If it passes that checkpoint, okay, it's going to receive the go-ahead to move into S phase and start reproducing its DNA. So how do cells work its way through that? Okay, there's internal as well as external controls on how cells move. So the internal controls are going to be proteins that are present in varying concentrations during the cell cycle, and these are going to decide whether or not we promote into moving forward or not. So evidence suggests that there are molecules that are present that cause a cell to progress from one to the other. So in varying experiments, let's say from S to G1 and S and S, when cells are in the S phase and they are fused with a G1 cell, they immediately enter S phase. Okay? So if you fuse these two together, they stay within that S phase. Cells that are fused from mitosis in G1, okay, immediately begin mitosis. Spindle forms, chromatin condenses, even though you haven't actually replicated the DNA, right? That vital S phase point. Okay, so even though that hadn't happened, it's still going to divide. So there's a protein that's called MPF, mitosis promotion factor, and then you have cycling-dependent kinases, cyclin kinases, and these that are important to promote the cell cycle, okay? Cyclin, right? So you can see that you've got fluctuations in these amounts over time when it's in varying levels and degrees of the cell cycle. So during mitosis, MPF is very high. During interphase, it's very low. You've got basically cyclin concentrations rise from S, rise through and kind of peak when it hits into mitosis and then drops very significantly until it's ready to move into that again. So cyclin-dependent kinases, this is present in sort of a constant amount. You've got the cyclin is on an on switch, so it's made in an increasing amount as the cell moves through. When you have cyclin and these two together, they turn on other proteins that's needed for mitosis, so like microtubule formation and all that jazz kind of helps with that. Now if you have no cyclin, you have no mitosis, okay? So I want you to think about the type of feedback that that represents, okay? So take a peek at that and stop, go back if you need to look at that diagram a little bit more. External controls, okay? Proteins and other environmental signals generated by other cells. In essence, hormonal type concentration, hormonal signaling, and things like that are going to be really important. So you've got platelet-derived growth factor, and so these are going to help with also causing cells to reproduce, right? So here is the idea of what happens. So you get scalpels, right? So a sample of human connective tissue cut up into small pieces, slice, slice, slice. Enzymes digest the extracellular matrix. You get a suspension of free fibroblasts. Then you transfer them into different vesicles. And when you get the PDGF is added to half the vesicles, okay, and when that happens, you're going to get more platelet growth, and you get, you know, and they create this growth factor that's going to help to promote cell division, which is kind of cool. So you get more cells grown when that's there. Now, why would platelets be important for this? Let's think about it. So when do you think about platelets? When do platelets ever come into a discussion? Clotting factors, right? So usually if there's a need for clotting factors, there might be a need for healing. Okay, so maybe you have a cut, right? And that's going to promote rapid mitosis to help to fix your cut that you might have on your hand, okay, and so on little wounds. So something to consider there. There's also positional inhibition. Normal animal cells have to be anchored, not too crowded. They're very density dependent. So when there's issues with that, basically, I got enough space. Space is, you know, if there's not enough space, they're not going to fill that space and they're going to stop. So there's a spatial awareness, if you will, that exists. Now, cancer cells, they don't have that spatial awareness. They don't care. And then they just sort of bleh everywhere. And that's why you get that sort of tumor that happens. So with cancer, this is uncontrolled cell division, all right? So, I mean, we know mutations happen. We know it's happening all the time. Your DNA is happening through entropic forces that are going to miss mistakes and so on. So we know the genes that make sure that mutated cells don't divide. But when those genes get mutated, okay, look out, then there's nothing controlling the division of the cell. So that is something that we look for, okay? So here's where these oncogenes come in. So you got proto-oncogenes. They stimulate cell division, the accelerator, as you know. So you've got your oncogenes. If they're mutated versions, they're always on. They never shut off, okay? And hence why you hear of people who, you know, doctors who focus on cancer, they're oncologists. So you're studying these oncogenes, otherwise known as cancer genes. So to promote that cell division, they're always, always on. And so when you have a proto-oncogene, okay, you get the translation, okay, gene moved to a new locus under, you know, new controls, okay, normal growth, okay. Gene amplification, when you get multiple copies, normal growth stimulating protein in excess, point mutation, normal growth, okay. If the point mutation happens in the oncogene, hyperactive degradation of the resistant, you know, degradation resistant protein, and you end up with the cell being locked into that cell division piece. Okay, so here's kind of one of the things. So growth factor comes in. Growth factor is a hormone, cell cycle pathway, because to grow, right, we need to make more cells. So growth factor comes in, acts at a receptor. Here is where you get hormone-mediated stuff going on, right? So it's a G-coupled receptor. So you got your G protein, which is a RAS, okay? GTP form of energy, you know, like ATP. We've seen these in in transcription, in translation. So RAS comes in, okay? You get protein kinase phosphorylation cascade, this chain of events that happens, okay? If there's a mutation, okay, and you have a hyperactive RAS, okay, which is because of that oncogene developing that, the product of the oncogene, the issues a signal on its own. You don't need the growth factor, because normally that hormone has to be the thing to trigger it, right? So the triggering of the hormone signals this. If you have a mutated RAS, it's going to trigger it without any additional growth hormone, because that's going to go through, you get transcription, the activator, you get the protein to stimulate the cell cycle, and the cell divides. So that's where you get some problems. Okay, and then we have tumor suppressor genes that are there to inhibit cell division. And if you guys recall, Sam Ryan talked about this. And it'll inhibit the cell division. It's like the brake pedal. Whoa, slow down that, slow down those that cell growth. However, in a mutated tumor suppressor gene, it's going to be off instead of on, and you're never going to cease that down. So here's some ideas of how that works when you have a defective or missing transcription factor, such as P53, and we will explore this in class as well, and what happens there, and it can't activate transcription. Okay, instead of inhibiting the cell cycle, there's no inhibition, and cell division occurs, right? And you get constantly going. So cancer is going to require mutations in these different genes to occur. It's very, very multi-step. The first thing that's going to happen as you go through is you get overexpression, increased cell division. You don't get the inhibition that you need. So you get the loss of the tumor suppressor gene, okay? Small benign growth happens. Then that's going to activate RAS oncogene, which is going to trigger more growth and more growth as it continues to become, and then if it becomes malignant, that's bad news and it will metastasize to other places. So this is an example of something like colon cancer. Okay. So this hypothesis here, this is based that the cancer occurs on average as a 6th function of an individual's age. Okay, so sort of the likelihood based on your age for developing cancer is going to be, obviously, the older you get, the more likelihood it's going to be. That's just that. Okay, so effects of the mutations. Protein overexpressed, cell cycle overstimulated. Okay, increased cell division. If you get an absent, if the cell cycle is not inhibited because the inhibitor protein's not there, increased cell division. So there's kind of twofold way to create cells that no longer are controlled. And that's bad news, right? So Gleevec, this is a cancer treatment. So to go back, I want you guys to take a peek at this and see this in action. Again, going back to the presentation. So stages of cancer, Again, I'm going to kind of pull back some of what Sam talked about earlier this year at the workshop. Various stages. You've got stage 1, 2, 3, and 4. Okay. And so in these stages, you've got the first one where you've got single cell, tumors growing from a single cell in one area. Pretty easy. Stage 2, cancer cells are starting to invade neighboring tissues. Not so good. Okay. Stage 3, now they're starting to, certain cells are starting to migrate to other parts of the body, and if they lodge somewhere and create a new tumor, that's stage 4, and that means that you're growing those cancer cells in another location from where they originated. And it's the metastasis part that is what's going to kill people, sadly. It's when it spreads, and that's what we don't want. So, on that note, hopefully we'll try to make things a little bit more uplifting. Make sure that you guys can do this. As we explore some of these things in class, we're going to kind of, you want to use this content to kind of help you with that. So how it develops, what the deal is with controlling, why it's so important to control our cell cycle, and what that means when it's not. So with that being said, have a great evening and or day or whatever time it is right now, and we will talk to you guys later in class. Take it easy.